Let's get started. Okay, so welcome to week 3 of Advances in Omics. So before I start, I have a couple of messages from Sir. So one thing, I think last time someone was asking about any textbooks available for these kind of the sequencing technologies and those kind of things, and Sir asked me to tell you that you can read the papers that are like attached with the videos that are shown in NPTEL. Those papers are very important, and also you can go to their websites and go through their documentation so that you can have a better idea on those. And the second thing is, I think I have not covered genome assembly in, I think, the first week. In the first week course, there was genome assembly covered. So what I'll do is I'll cover it today after I finish the threes part, and I'll cover a little bit of genome assembly. Okay, so let's get started. Yeah, welcome to week 3. So we'll start off with DNA nanoball sequencing. Uh, what is DNA nanoball sequencing? So basically, it is a technology which is developed by this company called Complete Genomics, and it was acquired by another company called Beijing Genomics Institute, and now it is marketed as MGI's DNBC platform. You can search for this term, and you'll be able to get more information on that. So, okay. So the idea of DNA nanoball sequencing relies on rolling cycle amplification, which is a method. Oh, sorry. Sorry, am I audible now? Yeah. Sorry for that. Okay. Yeah, today we'll start off with DNA nanowall sequencing. This is a technology came in around 2006, which was developed by this company called Complete Genomics, and it was acquired and commercialized by this particular company called Beijing Genomics Institute. Now it is known as NGI's DNBC platform. You can search on Google using this term, and you'll get more information on this. But the basic idea of DNA nanoball sequencing relies on this particular technique called rolling cyclic amplification. And this is used to create densely packed PCR-free DNA balls for massive parallel sequencing. Uh, so the rolling cycle amplification is basically central to this whole technology, and the idea is that you have a linear DNA. When you're sequencing, you have a linear DNA other than DNA like plasmid, which is circular in nature, but in general, human DNA or any other organism DNA will be linear. So uh, the idea is that you can amplify this into thousands of copies without PCR bias. This removes the PCR bias from it. So in other cases, even in Illumina, we use something called bridge amplification in Illumina, but in older technologies we use PCR to amplify the reads to before sequencing. So that introduced some bias into the reads, some PCR anomalies, and some kind of uh, yeah, redundant information got introduced in the reads, which while sequencing can bias your data. So this actually overcomes that particular need for using PCR, and it basically forms DNA nanoballs, which are around 200 nanometer in diameter, and this DNA nanoballs are fixed to precise spots on a chip, and cluster amplification is not required as done in Illumina. And the chemistry basically relies on fluorescent probes. I think by now you understand how these probes work. And these fluorescent probes and anchor oligos binding sequentially, and then you capture the image, and then you do a base calling. I think by now you are able to understand what these steps mean. Okay, so I just give a breakdown of the steps that have been created. Oh, so one thing I remembered is basically I told something during the start, but I was probably on mute. I should repeat that again. So firstly is basically sir has asked me to tell you that since last day some people were asking me for textbooks regarding these technologies and these kind of things. So sir has asked me to tell you that the papers that are linked to the videos, you can take a look at those papers. Those will be helpful for your understanding, and also uh, you can go to the publisher's website. So for example, go to Illumina's website, and so I've just opened one now. So here they actually kind of describe it in great detail. Remember, I showed you one video for the Illumina sequencing. You can go on YouTube and search for those videos. You'll get a better understanding of how these technologies work. Secondly, is basically I think from week one I have not covered the genome assembly part. So what I'll do is I'll cover it today in brief, and I think if you have any questions regarding that, you can ask me after this. Okay, going back to DNA nanoball sequencing. Okay, so these are the steps I've broken down into. So first is basically the library preparation. You break down the DNA into 200 to 400 base pairs, and then you have two adapters attached to the fragment ends, and which often will contain the barcodes and the sequencing anchors for anchoring into the plate. Excuse me. Then the whole concept revolves around this circularization into of a linear DNA into circular DNA. So DNA fragments will be ligated into single-stranded DNA circles using splint oligos and ligases, and linear DNA will be digested off to remove circularized molecules. The second step is the important one, which is the rolling circle amplification. A particular DNA polymerase is used, and the circular DNA will act as a tail plate, and the polymerase will continuously do the replicate around the circle. So you have a single strand of DNA. First you have a double strand, then you make it single strand, then you join it end to end, it becomes a circular DNA, and then you do replication inside the DNA, so it becomes a double stranded circular DNA. So it produces a long concatemer of identical copies end-to-end linked, and then this is what forms the nanoball. So the concatemer folds into a compact structure around 200 nanometer in diameter, and it contains about 300 to 500 copies of the insert sequence. So here the major idea is that you uh, do not have to use PCR, so there is no GC bias, and the signal intensity per nanoball is very good for imaging purposes and for the base calling purposes. And then after this is done, this the whole thing is loaded into patterned flow cells, and uh, they are loaded into predefined nano wells, and it ensures that one nanoball only enters in one spot to reduce the optical overlap during imaging. And it can go chips can be designed to handle up to a billions of nanoballs in one chip, so it becomes very efficient in throughput as well. So the final step will be the sequencing part. Yeah. The final step will be the sequencing part, which uses this technique called combinatorial probe anchor ligation, or CPAL, or modified sequencing by ligation. So it is similar to sequencing by ligation with some changes here and there. So the mechanism behind it is basically an anchor oligonucleotide would bind to a known adapter sequence on the nanoball, and a set of fluorescently labeled probes will be hybridized adjacent to the anchor, and a DNA ligase will join the probes to the anchor. And then it since the fluorophore has been added, you can image it and do base calling on those. And then again, this is basically uh, uh, it's basically you can the technique is you add a fluorophore, then you again remove. So this is reversible termination that we talked about in the last class. So then the fluorophores will again be cleaved, and the process will be repeated with the different position on the read. So on the right hand side is basically a schematic of how it occurs. So again, taking the DNA, making fragmentation, and you select certain sizes of DNA, and then you do end repairing and adapt ligation and sprint circularization. This circularization will be used to make different DNA nanoballs, and then you load it into the chip and to sequencing. So around, yeah, for the read length, it is around 100 to 150 base pairs, which is quite short in nature. And if it is a paired end sequencing, so basically you are sequencing from 5 prime to 3 prime and also 3 prime to 5 prime, so it will have around 150, 150, around 300 base pairs. And again, after that, you do imaging and base calling, and so basically you capture the fluorescence as you have done with other techniques as well, and then there will be a base caller software which will interpret the color and convert it to base calls, and the output will be in fast food. So the important thing to remember are a couple of things, which is basically combinatorial probe anchor ligation, which is CPAL, and the other is rolling cycle amplification. These two are the most important part of this particular technique. Okay. Moving on, another type is basically sequencing by IVDT. I'll not go much details into this, but this is basically, I think, uh, this paper. Yeah, I've taken the image from here only, so I will give you this particular paper. You can have a read if you're more interested in this. So yeah, this image is taken from that particular paper. Okay, so basically what happens is a reagent will containing multivalent avidite substrates, and an engineered polymerase will be combined with DNA polonies inside a flow cell. The engineered polymerase, so this is how it looks like. Uh, uh, then the engineered polymerase will bind to the free 3' ends of the primers template in the polyny and select the correct cognate avidite via base pairing discrimination. The multivalent avidite then interacts with multiple polymerases on one of the polymy to create avidity binding that reduces the effective KD. This is the dissociation constant of the avidite substrates tenfold, 100-fold compared to the monovalentite labeled nucleotide, allowing for productive binding of nanomolar concentration. So basically, you have a polyny of DNA inside your wells, and there will be a multivalent avidite molecule which will interact with certain polymerases inside the cell, and on one polyny it will create a binding with the polonies and the DNA polymerase and the avidite compound, which will reduce the basically the dissociation constant, and it will allow for a very high energy binding. So and then multiple polymerase mediated binding events per avidite will ensure a long signal persistence time. Imaging of fluorescent bound avidites enables base classification, and following detection, avidites are removed from the polonies. Extension by one base using an engineered polymerase incorporates an unlabeled blocked nucleotide. A terminal 3' hydroxyl is regenerated on the DNA strand, allowing for repetition of the cycle. So this is the same concept as the uh, reversible transfer termination of synthesis, where here also it's same that you have once that you have detected that then the avidite molecules will be removed from the polynase, and then again one base will be added to get back that 3' hydroxyl C group, and then you can again add the avidite molecules. So this is the workflow here. So you have these are your DNA polonies on these avidite will bind, and then you wash off the rest of the stuff, and then it will be specific to A, T, G, and C. So you can detect during imaging what base is present for what particular avidite has bound to what particular base. So you can uh, get this color, and then you again remove all the avidites, and then you get the polymerase, and you add uh, one blocking reversibly termination nucleotide, so that you can continue doing this stuff. And so in the next figure is basically how a single avidite molecule binds to a DNA polyny via polymerase mediated selection. I'll not go into the binding, but basically this whole technique and how the physics of this works, basically how the binding happens, and why it is so strong compared to other bindings. So this is actually all given in this particular figure and the paper. Please go through the paper if you want to have more information, but I just wanted to give you a general idea. So if you see the binding kinetics also, they have described in the paper, so you can give it a read as well. Okay, moving on. So the next part will be sequencing by binding, which is SBB. So this is the ONSO system by PacBio, which uses this technology, this chemistry, and it is basically high accuracy with Q40 level basis. So Q40 is basically the quality of base color assigned quality. So these are all Fred scaled. So basically, uh, Q40 means like 1 in 10 to the power 4 chances. So it is like 1 in 10,000 chances that it is wrong. So it's a very uh, highly accurate type of synthesis and sequencing by binding. So these are the steps that occur here. So first, the binding occurs, then the native nucleotide are presented with a polymerase forming a stable, non-cooperative tertiary complex with the template, and this binding event will depend on correct base pairing. Now, upon binding, again, similarly, a fluorescent signal will be generated and captured, and this will indicate the identity of the base without chemical incorporation. And after incorporation, again, when the fluorescent signal has been emitted, you capture it using some camera or imaging technique, and then here also unlabeled nucleotide will be added for regenerating the 3' hydroxyl link for the next cycle. So this is again an example of reversible termination of sequencing. So the advantage that it gives is basically the base calling fidelity is higher. So basically you have higher accuracy of translating of the signals to bases, and uh, especially it is useful for detecting rare variants and resolving homopolymer stretches. So homopolymer stretches will be basically repeat regions inside the DNA, and rare variants basically variants which are rare in the population, so you do not detect them very easily. So doing those is very useful by sequencing by binding, and also it incorporates scarless incorporation, which is basically use of native nucleotides to avoid chemical modifications, which is basically this step, the unlabeled native nucleotide incorporation, and it gets you highly reliable short reads, and basically uh, it focuses more on the precision than on the throughput. So if you want to detect something very rare in the population, or something that is very complicated, you want the precision more than the throughput itself, then this kind of technology is more useful. Like now, uh, before we move into long read sequencing, because the next part is long read sequencing, before we move into that, I want to I want to kind of highlight why we need long read sequencing. And I think right now the sequencing technologies are mostly for the short read, we use completely Illumina and similar technologies, but Illumina is the most widely used. And for long read, we use the main technologies out there are ONT and PacBio. So uh, I would want you to learn about long read from a perspective of why we moved from a shorter read technology to a longer read one. So, yeah, excuse me. Now, why is there a requirement of long read technology? Why do you need long reads for genome assembly, or like why do you need to sequence such long reads together? So that stems from the fact of the limitations of Illumina sequencing, right? So the read size, about if you do a paired read, so it's 150, 150, around 300 base pairs of paired read, and if you add insert sites, it's around another 150, 200. So it's around 400, 450 is the maximum read length that you can get for Illumina. Now, why is the shorter read length an issue? First, I'll tell you the advantage of short read. So, if you are sequencing a shorter read at one point of time, the chances of error are high, very low. But if you're doing it for a longer read, so here we are talking about 300, 400 base pairs, but in long read, we are talking in KB, like 5 KB, 10 KB is very common in long read. So when you are sequencing 500 base pairs together versus when you're sequencing 10 KB together, the error rate is highly variable, specifically for technologies like ONT. The error rate is quite high, going up to 5%. That is quite high if you are sequencing 3 billion bases at once, which is the length of the human genome. So, but still, long read has its uses. The major use of long read is basically detecting structural variants in the genome. So, I don't know if you remember or not. In the last class only, I think I gave you an overview about different types of variants, single nucleotide variants, and then you had the larger variants, which are greater than 50 base pairs. Those are denoted as structural variants, where you can have a deletion. Say, for example, you have had a 5,000 bases deletion in your DNA in a particular gene, which is actually contributing to some Mendelian disorder, some neurodevelopmental disorder, right? So identifying such a big portion of the genome has been deleted or altered in some manner is very difficult when you have reads of shorter length, right? So you can have multiple reads of shorter length, but it it is not encompassing the whole region of the genome where the alteration has occurred. So if you want to study structural variants, that is where the long read truly comes for helping out, and also it is very useful in kind of resolving highly repetitive regions in the genome. So when you have a lot of repeats, and if you have smaller reads aligning to those repeats, it's very hard to kind of align those reads because every in a space of 10 kb, every read looks the same because it's all repeats, right? So if you have, instead of that, if you have a longer read, which actually encompasses the whole region end to end, it's very easy to align that read to the particular genome. So these are the main advantages of long reads. So detecting structural variants, detecting repeats in the genome, resolving complex regions, which are the centromeres and the telomeres. We talked about T2T in the first class. If not, I don't remember if I had talked about or not. Uh, I'll just put the paper here. You can go through. Uh, so this is where they resolved the Y chromosome using longer reads. I think ONT was mostly used. And this is basically the GitHub page for the whole T2T consortium. If you need any resources, if you want to find reference genome for the T2T from the T2T consortium, you can find it here. And yeah, see here they have used 30x TagBio Hi-Fi, which is long read, 120x coverage of ONT, which is also long read. Again, TagBio. So these are long read sequencing, which has helped them. So the whole idea of T2T was to resolve the unresolved parts of the genome. So they used long read to do that. And the other publications are given here only. So yeah, this is the original one, the complete sequence of the human genome. You can read this as well. So before long read, it was not possible to kind of resolve the whole genome because you had a lot of very complicated regions in the genome, a lot of repeats, a lot of variations in the genome, which was not being able to detect using shorter Illumina reads. Okay, so this is the backdrop of why long read has come into play, and it's kind of, you can say, third generation, if you may. But yeah, so this is the kind of the latest technology, and it costs, long read obviously costs almost. Uh, I do not want to be misquoted, but it's, I think, around 10 times of what Illumina will cost. No, I think it has come down in the past few years, but it's still very expensive. So in consortium projects, in human genome projects like Thousand Genome Project or say for example, Genome Asia project. So in these kind of projects, they have used shorter read because when you're sequencing tens of thousands of individuals at one go, the long read becomes too much costly. And also the other issue with long read, as I was speaking, is basically the length of the read when it goes up, proportionately the error also goes up. So it is good for detecting structural variants, but if you want base pair differences, so single nucleotide polymorphisms, there, if the error rate is high, then long read will perform worse than short read because their per base sequencing accuracy must be higher to kind of get this particular readings, right? So yeah, so this is why long read is necessary. So the first type of long read that we'll be talking about is basically SMRT, which is again it uses sequencing by synthesis. So and the DNA is synthesized in a so-called, this is important, mode waveguides. These are known as ZMWs. So what are these? These are small well-like containers uh, present in kind of a plate where there are a lot of wells. So these are well-like containers, and inside each well, the sequencing occurs. The sequencing is performed with the use of unmodified polymerases and fluorescently labeled nucleotides flowing freely into the solution. The wells are constructed in a way that the fluorescence occurring at the bottom of the well is well detected. So you capture it from the below, not above. And the fluorescent label is detached from the nucleotide at its incorporation into the DNA strand, leaving the unmodified DNA strand. And the SMRT, sorry, the type here, and SMRT technology allows for detection of nucleotide modifications, and it can happen through the observation of polymerase kinases. So I don't know how much biology you guys are studying, but do you know what kind of nucleotide modifications can occur in the DNA? Any idea about that? And why do you want to detect that? You can write in the chat what do you think. What kind of DNA modifications would you be looking at when you're sequencing? Okay, okay, I'll yeah, correct. Yeah, DNA methylation. That's correct. So we are looking for epigenetic modifications, right? So what will happen? What is happen is uh, you have the DNA. The DNA is not only the nucleotides, right? So you have certain. So how is the DNA packed? The DNA is packed. The DNA is negatively charged, and you have histone octamer proteins which are positively charged, and the DNA is kind of wrapped around these histone octamer proteins. Now, where higher concentration of these histone octamer proteins will lead to what is known as heterochromatin. So those are densely packed regions of the genome, and there are also loosely packed regions of the genome, which is known as chromatin. Now, in the heterochromatin part, if you need to do replication, say for example, the cell needs to do replication in the highly packed region. How will you do that? You first have to unwrap the DNA from the histone octamers, and then you do your replication or your transcription, what you want to do. So these wrapping, unwrapping, and where this particular uh, DNA replication or replication or transcription will start, and those kind of things are controlled. So if you can control when this DNA will be unwrapped or wrapped, you can control the gene that is going to be expressed along this region, right? So that is controlled by certain modifications on the DNA, mostly methylation on the DNA and also acetylation on the DNA. So there are a couple of enzymes which are HMTs and HCATs, which are histone modified, histone methyl transferases and histone acetyl transferases. So these kind of enzymes actually handle this wrapping and unwrapping part, and where and when and how this transcription will occur. So finding these signatures on the DNA, right? So if the particular region of the DNA is methylated or non-methylated, that can tell you a lot about the structure of the DNA in that region, in terms of how it is wrapped in the around the histone and how accessible the DNA is actually. So which you cannot get from a direct just sequence extracting the DNA and just sequencing the DNA to get the nucleotide. If you have a little bit more information on if the methylation has occurred or not, and what these methylation signal actually tend to say, then you can have a better understanding of how the DNA is behaving in that region, right? So epigenetics is very important. It is as important as genetics because uh, epigenetics will control the first step of how and when replication and transcription of the DNA will occur. So that is very important to understand. So again, it allows detection of nucleotide modification, and this is kind of true across long read technologies in ONT as well. You can detect these modifications directly while sequencing, and this happens through the observation of polymerase kinetics, and it is slightly higher. The read length is slightly higher, but we are not there yet. It is around 1,000 nucleotides around that kb. Okay, so this is a schematic of how it looks. So you have the nucleotides with fluorophore signals, fluorescent signals attached, and then you have this is the DNA polymerase, and it is adding. This is basically the primer, and then it is adding each molecule one after another, and it detects the light signal from somewhere here, and when the signal is added. So yeah. We come to another type of long read sequencing, which is hi-fi reads using PacBio sequencing. So this is actually one of the kind of the widely used technologies now. Yeah. So this is the PacBio sequencing. So you go to PacBio hi-fi. Yeah. So hi-fi stands for high fidelity, right? So you can watch the videos and basically understand uh, more in detail. So you can follow these basically the websites to understand the technology in greater detail. Anyway, so here what happens is basically these hi-fi so-called hi-fi reads are basically produced again. The concept is similar to kind of circularizing the DNA. So these are produced using circular consensus sequences, which is CCS mode on PacBio long read systems, and this hi-fi read produce a base level resolution of 99.9% single molecular read accuracy. So basically, what happens is you start with a DNA, and then what you do is kind of add linear DNA, which actually binds to both the strands. So on the end uh, it becomes circularized like this, and then what you do is basically you cleave off these bonds and do a double-stranded kind of synthesis on this using polymerase, and it becomes a circular DNA. Right? And the polymerase reads are then will be trimmed and to yield the subreads, and the consensus and the methylation status. Again, the DNA modification, the methylation status are called from the subreads. So the idea is similar here as well. So you have well, like you have basically plates on which these certain bases will be added. So when this base will be added, it'll emit a signal which will again be captured. But the unique thing here is basically you are circularizing a part of the DNA. We are not cleaving it, by the way. So you are only circularizing a part of the DNA, and then you are trying to sequence that part, which basically helps you a little bit more with the accuracy of the reads. So here also, I think hi-fi I think gives you uh, around similar as ONT, I think around 5 to 10 kb as N50. N50 is basically the median length of the reads across the genome. So, yeah, I think that is, I do not recall completely, but it is closer to ONT. But yeah, the important thing is the circular reads actually help you reduce the number of errors while doing the base calling, and so it is important for variant detection, epigenetic characterization, and you can also use it in RNA sequencing and other technologies as well. Yeah, that's fine. Okay. Next we come to the Oxford nanopore technology, which is basically another long read sequencing technology. But here the idea is slightly different. Here you detect the bases not using fluorophores, but the change in the flow of current. So how does it work? So you have uh, uh, basically an kind of a nanopore molecule. So this actually looks like an S0F1 ATP synthesis, if you guys know. So basically, you have two molecules, one on top of another, and there's a pore down the middle through which the DNA actually passes through. So one protein will unzip the DNA helix into two strands. So you have two double-stranded DNA. One protein will do the unzipping, and the second protein will create a pore in the membrane and holds the adapter molecule. So here is the adapter molecule. So I don't know if you can see this. So here is the adapter molecule, and basically the second uh, molecule, meaning this particular molecule, will hold the DNA in place, right? So and while this is the state, so you have an adapter molecule. This is holding this DNA in place, and this particular molecule is kind of unzipping the DNA. At this stage, what they'll do is they'll flow ions through this pore, which will create a current, and each base will block the flow to a different degree, altering the current. And that is how a particular base will be detected, because a particular base will have a clear signature of how much current it will allow or it will resist. So by that information, you create a signal graph and you can understand. So, for example, G will kind of say increase. So from T it is kind of gone down, then G it has gone up. Again, if you see at this position, T has gone down and G has gone up here. So at all these are basically signatures. This is not too kind of detailed in that manner, but the idea is that each base will change the current, the ions flowing through the pore, basically the current that is flowing through this pore to a certain degree, and to what degree it is changing will actually determine if what base is particularly handed over here. So you, for each base, it can detect the change of current, and that is how it will detect what particular base is there. Right. I don't know the image is pretty bad, but this is actually from the original website as well. You can even visit it and check it out. So first, a nanopore will be inserted into an electrically resistant membrane, and then a potential difference will be applied across the membrane, which will result in a current flow through the aperture of the nanopore. And a single molecule that enter the nanopore will can cause characteristic disruptions in the current, and measuring that disruption, the molecule can be identified. So this is the background technology used for ONT, right? So I earlier used to detect using changes in color of the fluorophore. With ONT, you are detecting by change in current. Okay, so again, there are a lot of pros for doing it this way, getting a longer read. So yeah, so the minimum amount of data processing and complex algorithm are required. So it's very, in that term, it is simple, and that we can identify the genomic sequence at large scale at one go, because the read length will be very high. The size of the pore is fixed, does not vary for large variations of environment or experimental. Real-time sequencing at high rate of accuracy of about 98%, but again, ONT is known for some being a little bit more error prone. So be a little careful. 98% meaning you still have 2% of error, and 2% of 3 billion is a high number, right? So you have to keep that in mind. So yeah, the cost is slightly has come down now. So it is around $1,000, I think, but still I think it has come down from that as well. If you do it in bulk, I think you can do it in less money. Translocation of polynucleotide through the pores are precise and controlled, because you have that adapter sequence in the lower molecule, so that it can perfectly hold the molecule while the current is getting passed. So basically, the accuracy, the base that you'll be sequencing will be very precise and controlled. But again, there will be cons. So the first one being the DNA sequence analyzing system is yet to reach its maximum efficiency. So we are still developing this technology. We're not there yet. Having a long strand of single-stranded DNA is also a major concern to avoid self-complementary binding, and this can actually, so if you are basically using a single-stranded DNA to uh, passing through the pore, so it does not always work well. So you can have self-complementary binding, and this whole thing can fail. So you have to be very careful at those steps. And though it can work in various experimental conditions, it has some temperature limitations because again current is flowing through the spore, so it has to be in a very controlled environment because the current flow will determine what base is getting called. So if anything that hampers the current flow or the ion discharge, that will hamper the base calling. So that is very important. Again, high signal to noise ratio is yet to obtain. So you have a lot of errors and a lot of noise. And also the number of run per pore membrane is also a bit low. So yeah, it's not massively parallelized, but you have longer reads. So that is the important thing, that you can have such longer reads that it can kind of uh, negate these cons. So yeah, it's right now it is the state of the art technology. We do not we have certain technologies that are coming up, but widely used state of the art is right now ONT and PacBio. Both of them. Long read in general is kind of the state of the art, but still there are the con with long reads is basically at single molecule level accuracy where the error rate is higher than say Illumina sequencing. So still Illumina has its place to when we are doing large scale projects or large scale population genetics projects and those kind of things where you're sequencing a large number of individuals together. I think still Illumina is a better option, and when you're detecting single nucleotide variants, still shorter read is a better option than longer reads because of the high error rate of uh, longer reads. Right. Okay. So this part concludes the this week's week three's all materials. But again, I at the start of the lecture I told you I'll be talking a little bit about the coverage because I've not covered that. So I'll go to that part now. Okay. Okay. So some of these slides have been kind of taken from the internet from Professor Ben Langmuir's lectures. And so he's a professor at Johns Hopkins University. He teaches, I think, in MIT also, I think he teaches. So I have taken a couple of his slides here to explain. I think if you want to, uh, yeah, you can take a look at his teaching material as well. So there's a lot of resources in his teaching material. So I've taken a couple of these materials from here. Okay. Okay. Uh, moving on. Okay, so we'll talk about coverage and assembly, but firstly, we need to define what coverage is basically. So coverage means a kind of coverage refers to the number of reads covering a particular position in the genome. What does that exactly mean? So this these are all the reads of this particular region of the genome. So you have to imagine there's a reference genome to which you're trying to align these reads, right? So by that, when you're doing that alignment, you'll see overlapping reads, right? So uh, a single position will be sequenced multiple times because of the PCR amplification or the bridge amplification or the read amplification that you have done and you have sequenced those reads. So you have sequenced different regions of the genome multiple times, right? So how many times you have sequenced and how many reads actually are getting aligned to that particular base will tell you the coverage of that location. So in this case, this particular position, so this is the reference genome here, and these are all the reads that are aligning to this region of the reference genome. And this particular region, this particular position right? This particular position is given a T by six such reads. So six reads are supporting that there is a T in this location, right? So this that is what is known as the coverage. So coverage of a particular base is basically how many times a particular base is supported by how many reads in that particular region. So in this case, there are six reads supporting that there is a T here. So that is what the coverage is. Now, what is assembly? So genome assembly is basically the the idea behind genome assembly is basically the most similarity there is one end to another, and so basically if you take two reads, this and this, you see a lot of the read is overlapping except for this particular region. So the principle is that if you have such overlapping reads, the idea is that they might have originated from the same region of the genome, overlapping stretches of the genome. So this is the particular reference genome, and then you have overlapping reads like this. So this particular, so when you do not have the reference genome, what you can try to do is you have this overlap between the two reads, and you can try to construct the whole genome from this just the overlaps, right? So you have multiple of these reads that will be overlapping one after another, and by matching these overlaps you can construct the whole genome. So for example, if you just take these two reads, the region of this particular DNA will be read out as TCTA, and then this part is fully common. There's one mismatch here. There can be mismatches, we'll come to that. And after that, a CC at the end. So TCTA, this whole set of nucleotides, and then CC. So that is what the principle of uh, assembly is, that you do not know uh, what is your target genome, but you can get the target genome by checking the overlaps between the reads that you have sequenced. Right? Now, say for example, two reads truly originate from overlapping stretches of the genome, but there might be differences. As we saw here, that there might be differences. Now, why is that the case? Firstly, that can be sequencing error. Secondly, there can be differences between inherited copies of the chromosome. So basically, when in a child, one copy of the chromosome will come from the one parent, the other copy will come from the other parent. So the copies might not be exactly same, right? So there will be variations in the copies, and if that is the same, then in diploid organisms like humans, um, these copies might differ. So this uh, difference might be due to that, because you are taking the first read from the mother and the second read from the father and trying to align it, but there is there can be minor differences here, or it can be as simple as a sequencing error. So this is the sequence from the mother, and this is the sequence from the father with one change in a base position. So this is actually a variant in the genome. We'll come to that later. Now, as you saw, finding overlaps is very important, right? So when you're doing a genome assembly from scratch, you have all the reads together. How do you build the whole genome? Finding overlap between the reads is very important. So what we do is basically find all overlaps and build a directed graph where basically each of the nodes will be each of the reads, and the edges connecting between two nodes will be the overlapping stretches of the DNA. So for example, you have these many reads, and you want to connect one read to the another. So basically, this whole read actually overlaps this read like this, right? So each of these node will represent a read, and one vertex, one yeah, nodes and vertices only. So the one vertex connecting one node to the another will represent the amount of overlap between these two nodes. So again, these two are independent nodes. Why they are connected? Because there is a overlap between them. So mathematically, this is how we represent. Because again, we are talking about billions and billions of reads, right? So manually it can't be done. You need to have efficient computational algorithms and models to kind of do this. So mathematically, this is how it is represented. So this is a directed graph. So the arrow goes from this to this. So basically, the suffix of the first node is overlapping with the prefix of the second node. So that is the that is told by the direction of the arrow. Right. So this is the first node. The suffix of this node, basically this part, is actually overlapping with the prefix of this node, which is actually this part. So this arrow tells you that the direction of the graph comes from that concept. So again, just a quick uh, recap if you are unfamiliar with graph theory. So basically, in a directed graph, you have a set of nodes which are connected by edges which are directional in nature. So it starts from one node and it ends on another. So one is called the source, the other is called the sink. Right. So I'll not go very details into that. That is probably all you want to know. Okay. Now, how do you create an overlapping graph? Because again, you have these reads. Now you want to create the whole genome. You want to need to find overlaps between the suffix of one read, prefix of another. So this is how you create an overlapping graph. Basically, then an overlap will be the suffix prefix match of at least three characters in this case. A vertex is a read, a directed edge is an overlap between the suffix of the source and the prefix of the sink. So these are all your vertices, basically your nodes, and these are your edges, which are your overlaps. So this is how it will look like. So this is your first node, second node, and third node. These are three different reads, and this is how it is overlapped. So if it is overlapped by three positions, the weight of this particular edge will be 3. If it is overlapping 4 nucleotides, the weight of this edge will be given as 4. So we also give weightage to these edges to signify the amount of the overlap, how close these reads are together. Right. So and it can also contain circles. So A to B to C, and again C can go back to A. A cycle path is the beginning and the ending at the same vertex. This can happen when, firstly, when the DNA string is itself circular, for example, in bacterial plasmids, etc. But more importantly, the circles can be due to repetitive DNA, because again, there will not be very much differences between these three reads because these have originated from the same repetitive region of the DNA, and these are very much overlapping. So if you have repeats in the reads, the finding overlaps between these repeated reads are not going to be very difficult. So it is very much possible you can have recyclical uh, overlap graphs, right? Now, how do you find this overlap computationally? So, say for example, you have a length L suffix of X match and length L prefix of Y match when L is given. The idea is to look in Y for occurrences of length L suffix of X. And so these you extend these matches to the left and the right of the column where the entire prefix of Y matches. So first you take these are two of your reads X and Y. So you are looking for this the suffix of X in the prefix of Y. Right. So from for the first street, you go from right to left. So you start up from the suffix and you start going from right to left. And from Y, and you try to find overlaps with Y. So in this case, GCC is kind of overlapping here, and then TAG is kind of overlapping here. So then you once you have certain overlap, then you kind of go extend towards the left and to see if you have particular overlap. So now you can see how these two reads are overlapping. So you start from the end, then you go to the left, so CCG, then you try to find where this CCG is in the second read. It is somewhere here, CCG, and then since CCG has matched, you move a certain left to match the next three nucleotides. So this how many nucleotides will match is given by this parameter L, which you can change, right? So in this case, we are considering three. So first the GCC has matched here. Sorry, CCG has matched here. Then you look for GAT. Then GAT, then it matches here, right? So to take this L depending on your computational algorithm, and then you try to find the suffix prefix matches like this. Now this is if you do it, do a small string and kind of break it down. So this is basically the original string, and then you have broken it down into smaller reads, and then you try to find how the graph is looking for these reads and how they are overlapping. So this is an example overlap graph with L equal to 3 overlaps minimum, and each edge label will tell you the overlapping length. So for example, you start from here, you go to here, that means three nucleotides are overlapping. TAT is overlapping with TAT here. Again, if you go from this node to this node, six nucleotides are overlapping. So like this is how, and or if you have an original string and you break it down into reads, and this is how the overlap graph will look. and this is a first step towards the genome assembly. Right? Now, computationally, this is known as the shortest common substring problem. The idea is very simple. You have been given a collection of strings and you are told to find the shortest common substring among these strings. The shortest string that contains all the strings in S as substrings without requirement of shortest is easy. You basically just concatenate. So basically, if you're given this set of uh, substrings, you can say you are asked to make the shortest common substring. If you are not asked to make anything shortest, you can just add them, right? It'll be a read. But if you want to make a shortest common substring, it becomes much shorter like that. So if you just add it, it'll become 24. The length of the read will be 24. If you want to find shortest common substring, like one after another overlap, then it becomes 10. So you see the difference. You cannot just add up the reads, right? You have to find these overlaps and then find the shortest common substring. So that is the computational problem that we face. Now, if you imagine a modified overlap graph where each edge has a cost, which is equivalent to the negative length of the overlap. The shortest common substring corresponds to a path that visits every node once, minimizing the total cost of the path. So that is known as the traveling salesman problem, which is very common in graph theory, but it is NP hard. It is computationally, it is not feasible to solve this. Now, say for example, we disregard the edge weights and just look for a path that visits all the nodes exactly once. Right? So you start from any node, and so the traveling salesman problem is basically you start and in one node, but you have to visit every other node. You have to minimize the total cost of along the path. That is computationally very hard to do. It is NP hard. But if you try to disregard even the edge weights and you just look for a path that visits all the nodes exactly once, that is known as the Hamiltonian path problem, which is NP complete. Uh, uh, it is and it is well established that this finding of the shortest common substring is NP hard, which is very difficult in a computational algorithm. Now, yeah, this is a greedy algorithm. I'll not go into the algorithm details because this is not a computer science course. But again, if you want to learn more about this, this is like big data algorithms. You can again go look into Langmuir's lectures. There are other people also who deal with this algorithms for genomics and other uh, kind of big data algorithms for biology, right? So yeah, again, this is how the greedy algorithm will look like. You have a set of input strings, uh, and then you are trying to find the shortest possible superstring. The idea is that non-optimal superstrings can be found with a greedy algorithm, and at each step, the greedy algorithm will choose greedily the longest remaining overlap and merge it with the source and sink. So it'll just check, it'll first arrange it and then check the maximum overlap, and then it'll just keep on arranging. So for the if you do the greedy algorithm, and with all the strings, you get something like this, which is the superstring. And this is the answer for the greedy algorithm, but the actual shortest common substring is at this. So again, greedy algorithm is not guaranteed to choose overlaps, yielding shortest common substring, but it is actually a good approximation. If you look at it, it might not look the same, but there are some elements, say for example, three B's that are one after another that is also represented here. So there will be some kind of uh, ac- not accuracy, I'll say some correctness to the answer, but actually it's not correct fully. It's a good approximation, and the superstring algorithm by superstring yielded by the greedy algorithm won't be more than 2.5 times longer than a true shortest substring. So that you can prove this mathematically that even if you use a greedy algorithm, it will not be like 2.5 times of the total of the actual answer, right? So, but in case of genomics, we are talking in billions of bases. So that becomes 2.5 times is a very big number for that. So it becomes harder. Right? So, so you use these overlap graphs. So you build these overlap graphs and build a layout. bundle sketches of the overlap graphs into contigs and pick the most likely nucleotide sequencing for each contig. So again, it's the same concept we talked about here. You, you have X and Y, these are your two reads. You start from the right and move to the left to find every pair of these overlaps. Right? So then what is built is known as a suffix tree. So basically you have you build what is known as a suffix tree. So these are all your nodes, and the overlaps will tell you basically how what are the strings that are getting overlapped. And say for example, uh, you go to the first string is basically GAC ATA. So this is the first string. So GAC, you start from this is the root node, and then you move to the other nodes. So GAC ATA, this is your first, this is your query. You have to find where in the uh, whole of this suffix tree, because you have made this suffix tree of all the reads that you've gotten. Now you have to find where is this particular string in the suffix tree. That is your query. And so what you do is basically you start from the root, you follow a path which actually tells you GAC, then ATA. Then the green edge length tells us the length 3 suffix of second string equals length 3 prefix of the query. So basically, what it means is the length GAC and then the second node that you're going into, either ATA or the second node. It basically you are following a path, right? You're going from a root node to another node, and then you're seeing what are the paths possible, and you're following those paths. So it will tell you if you go by the green edge, it will be like 3 nucleotides less. So the obvious way to go will be the red path. So again, these dollar signs, do not get confused by this. These are basically denoting ends of the string. So this is basically a generalized suffix tree for just these two reads. Now imagine you have like 150 base pairs read. I'm talking about Illumina, 150 base pairs read, and you have billions of these reads, and you have to make something like this and then query. So computationally, it is very intensive. So the idea for assembly, the strategy for assembly is basically first to build the tree, and for each string, walk down from the root node and report any outgoing edge labeled with a separator, and each will correspond to a prefix-suffix match involving a prefix of the query string and the suffix of the string ending in the separator. So I'll not go into the detail of this. So this is basically how it works. You start from the root, and then you try to follow certain paths. So and you try to find what the string is. So now try to think physically what this is doing. So starting from the node, then moving to other nodes from the root. It is what you're doing is you're trying overlaps. So because this is an overlap graph, so you when you walk from this node to this node to this node and this node, and when you complete this path, and then you put all the overlaps together, you find the whole string from the root to the leaf node, right? Yeah. So that is how it works. Again, I'm not going to go into the uh, details, but yeah, this is how it looks like. So this is actually linear in time. So time build time to build a generalized surface tree will be o to the O(n). So it's not O(n) square and those kind of things. So it's a decent time complexity, right? Yeah. So again, you can do this with find this overlaps with dynamic programming as well. So again, I just want to give you an idea. I do not want to go into details because this is actually out of the scope of the course. But again, I wanted to give you an idea about genome assembly. So okay, but this I'll talk about is what is known as De Bruijn graph assembly. So this is a very widely used technique to find these overlaps and find the whole string. So there's this concept of K-mud, which you'll also hear a lot in terms of genome assembly. So K-mud is basically a substring. It's nothing. The K stands for the number of strings in the substring. Basically, a 4-mud means basically 4 nucleotides containing substring, and 3-mers is basically 3 nucleotide containing substring. Right? So this is what K-mud means. Mer comes from the Greek word meaning part, and we use k minus 1 mer to refer to a substring of length k minus 1. So k is basically length of the substring. Okay. Now, basically, you have, say, for example, the whole genome, and then you break it down into small k-mers. So that is what we are doing while sequencing, right? So we are breaking it down into small k-mers. Now our problem is to bring these all k-mers and get the whole substring, whole string from it. So again, say for example, these are your k-mers where k equal to 3. Uh, AAB is your k-mer. A is the k minus 1 mer, and AB is the k minus 1 minus, sorry, A is the left k minus 1 mer, and AB is the right k minus 1 mer. So this is how it is divided, left and right. So now if you take each length 3 input string and split it into two overlapping substrings of length, uh, say 3, and you call them the left and the right two mers. So basically, you get a string and then you divide it into all the substrings. These are basically all your reads. Now, inside all these substrings, all these three mers, you break it down into the left k minus 1 mer and the right k minus 1 mer. And the and these two mers, basically, since your k mer is 3, the left and the right will be k minus 1, so it will be 2. So let the two mers be nodes in a new graph, and then you draw direct rays from each left tumor to a corresponding right tumor. So basically, you try to connect each of these smaller k-mers, and then each edge of this graph will be corresponding to a length of 3 input strings. So basically, what this means is basically AAB. Since there's one overlap, then ABB, there's one overlap here because you've broken it out into right and left, right? Okay, now when you have done all these overlap graphs and kind of breaking down into smaller k-mers and building this graph, now you have to get the whole string, right? Mathematically, this is what is known as a random walk. So what it is basically a random process, statistically a random process, which describes a path that consists of a succession of random steps on some mathematical space. In our case, a random walk meaning you start in a node and then you try to move to other nodes depending on the connections, right? And at each node, you have, say, for example, a node will be connected to three other nodes. So depending on the edge weight, again, remember edge weight is basically the overlap between two k-mers, right? Depending on the edge weight, you'll have some probability of moving to one node versus the other node. So the idea is the given a graph and a starting point, you select a neighbor at random, move to the next selected neighbor and repeat the same process till the termination condition will be verified. And the random sequence of points selected in this way, it's what is known as the random walk of a graph. So I'll just quickly give you an example. So this is what random walk looks like. So iteration 0, you start with node 1. So your probability of being at node 1 is 1. Right. Now, if you want to move from node 1, you have two options, node 2, node 3. So in the next iteration, the probability of going into node 2 and node 3 becomes 0.5, 0.5. Right? Now, when you have moved to node 2, the moving, you can either go back to node 1, you can either go back to node 1 or node 4. So you can calculate these probabilities, and the path of the highest probability will be basically your best fit path. So that will give you the best string consisting of all the substrings that are present in this node, right? So I'll not go into the detail, but this is how it works. So you start at the node and you try to move to other nodes depending on the probability of moving into the node will be proportional to the edge weight. So, and in this case, it is given by the degree, the number of connections of a particular node with other nodes. So a, a continuation of random walk is what is known as Eulerian walk. So uh, the assumption in Eulerian walk is basically a node is balanced if in degree equals out degree, and the node is semi-balanced if the in degree differs by the out degree by 1, and the graph is connected if each node can be reached by some other node. The idea of Eulerian walk is basically you visit each edge exactly once, not the node, but each edge exactly once. You can go to a node multiple times, but you can travel from one node to another in a particular edge only once. Right? Not all graphs will have Eulerian works, but graphs that do are known as Eulerian. A directed connected graph is Eulerian if and only if it has at most two semi-balanced nodes and all other nodes are balanced. So yeah, again, this graph is actually Eulerian. And so the idea is the walk is known as Eulerian walk, and the graph that we build is basically a De Bruijn graph. So for a genome, it looks something like this. So for example, assume perfect sequencing where each length k substring is sequenced exactly once with no errors. And you pick a substring of length k, say for example this, and then you break it down into smaller substrings. So this particular line will look something like this. And then you start with an input string, take each k-mer and split into left and right k minus 1 mers, add k minus 1 mers as nodes to the De Bruijn graph, and add edge from the left k minus 1 to the right k minus 1. And then you start at one edge, and then you do this Eulerian work, basically not visit, you can visit one node multiple times, but you cannot travel one edge multiple times. So that gives you, that is that mathematically and computationally how genome assembly works, right? Uh, yeah, that is all from my side. So I know this. I've talked a lot in terms of the computation and this kind of suffix trees and suffix prefix matches and shortest common substring. Don't be kind of overwhelmed with that. But I just want you to appreciate the kind of computational power and the mathematics needed to do something like this, which is uh, in terms of if you just do it in a plain simple finding shortest common substring, it is basically NP hard. So from that we have come down to better algorithms, better computation and mathematics to kind of understand this genome assembly and kind of uh, do it in a much more efficient manner. So all the details are not maybe not that important, but I just wanted to appreciate uh, the kind of effort that goes into doing something like this, both computationally and in chemical manner. So all of the chemistry is very important while sequencing, but after sequencing, the job is not complete. You need a lot of mathematical and computational expertise to kind of uh, build these graphs and get the whole genome and do alignment and do variant calling. A lot of computation and mathematics is required there. So, uh, yeah, I hope you liked it. If you want further information, as I said, Ben Langmuir's lecture notes are very nice. He speaks a lot about these algorithms. These are what are known as big data algorithms, and yeah, so you can take a look here. And he also, I think, has an MITx course in YouTube if you want to learn about that. Okay, so we'll move on to questions for today. Um, if you have any questions, please ask, or else we'll move on to the assignment questions. Yeah, I'll go through the assignment. So yeah, let's do that only. Okay. Assignment one. So I think did you attend? Just a second, Swati. Did you attend last day's session? I think I discussed the issue with that particular problem with contigs and gaps, APGP. Are you talking about that? Yeah. So that I talked with sir. So that was kind of a tricky question. So they have asked you for the genome assembly size, not the genome size. So there's a technical difference between the term, right? So genome assembly size is just the addition of the length of the contigs, not the gaps. The gaps are not considered in genome assembly size. When you're doing genome size, then you consider the gap as well. So in that case, you just disregard the whole um, gap information uh, given in the question, and you just use the, I think, the contig length wherein GP, if I'm not wrong. So you just add the contig length depending by calculating the GP. I think you'll get the answer. I think it's the first answer, not not none of the above, or like around 260 KB that we are getting. That was wrong because we were adding up the gaps. So you do not have to add the gaps. They have asked for the genome assembly size, not the genome size. So you will need to know the total number of contigs, but you can get the total number of contigs using the GP formula. I do not have that question right now, but you can get that using the whole using the GP formula. You just do not need to consider the gaps. You just ignore the gap information that is given to confuse. I think the total number of contigs were given. Let me check once. I think. Yeah, I think the longest was given as 131 KB, right? From that, you can calculate the total number of contigs. You do not need the total number of contigs. They have given the longest length of the contig, and basically the shortest contig also they have given 2 KB, I think, and they have said that they are in uh, GP. So you can use the GP formula to find n, basically the number of contigs. I think the answer is the first option, 174762. Okay. Is this from this week or earlier weeks? Okay, which one? Okay, you can do one thing. You can write the question in the chat box. Just let me check once then. Question number 14. Yeah, I think the answer is TRN. Why do you think it's not a genetic material? Meaning, like, I do not exactly understand what you mean it's not a genetic material. I think you are talking about from the point that it does not code for any protein or anything like that. I think you're talking from that perspective, but yeah, so I don't think that question was that deep. So it's, I think it'll be TRN. You see, I do not make these questions. So if you have any issues with the questions, you can directly write to Sir in his email ID. But yeah, some questions might be like this, like it means something, and like it does not actually mean what you're thinking. So if you have any like these kind of issues, you can email to sir as well. So I think he can help you, because there's another TA actually who makes these questions, and I am not that person. So I just my job is to kind of discuss this question and the answers, and basically whatever has been uh, uh, kind of covered in this week's course. So regarding if you have any qu- like framing of the question issue, I think you should directly mail sir, or I can also do it on your behalf if you want. Maybe in the discussion forum also you can bring this up. Okay. Yeah, yeah. Uh, Lipika, I think that was the one I was talking about. So I went back and checked the question. So basically, they have asked for genome assembly size, not the genome size, right? I don't know if it was the 16th question or not, but I think you're talking about the same question. So there, you do not need to consider the gaps. We were considering the gaps, and that's why we got around 260 kb, I think the answer. So we discussed here none of the above, but I think I went back and checked. So the gap information is given to confuse you. You just need to not consider the gap information. If you just add up the contact lengths, it will come up to that 1717476 kb. I think, yeah. I think I discussed this last week as well. So yeah. Okay. Okay, here the answer is basically the CPAL technology. So after they acquired complete genomics, they actually started using the sequencing using the CPAL technology. So that was the technological change made there. Yeah. Okay. Okay. So both of thing, both of the things can happen. First is if you remember. So this is how it looks like. Where did you go? Yeah. So you can independently add because the avidite will independently bind to the bases, right? So you can do both things. You can add it all together, or sorry, or you can detect the base and then you can independently add the ability as well. So, but these are independent things, so you can do it both. But you cannot do later because if you do it later, you miss out on the signal for that particular base. So either you do it simultaneously, or you do it and just at the moment base is detected and then the next is added. Right? So later you cannot do. Yeah, that's on me on. Uh, okay, before I forget, I think before we move on to the next question, I think I thought I'd show you the consortium projects. So I think Sir discussed it is 1 KGP, 1000 genome project. So this is actually, these are the publications, right? So if you want to go through what has been done, I'll just open one of them and show you. So basically they sequence this 1000 individuals from different ancestries and what kind of variants they found. So this is the pilot project already described, different ancestries that we have taken, how they have done variant discovery and this kind of thing. So all the papers, so these came out in series. So already I think there are 4 papers in Nature for 1000 genome project. These are the publications if you want to take a look. And if you want to like use the data, that is also possible. So you go to, so this is the home page. So the International Genome Sample Resource for 1 KGP. Then you go to data, then you scroll down and you can go to the FTP site, right? In the FTP site, you can take any phase. I will go with phase 3, go to the data, and these are all the samples, right? These are the sample IDs. Go inside any of the sample IDs, and get the sequence read, you get the FASTQ files right from here. In some cases, you have the BAM files as well, which are the aligned files. So you can take these files, FASTQ files. These are actually human genome data, right? Uh, so you can, if you want to play around with the data, use it in some pipelines or something like that. So it is fully available. So if you want, in some cases, BAM files, in some cases, FASTQ files, for example, in this case, you have alignment files as well. These are your BAM files. So you can use these as well. So the 1000 genome project is a very interesting one. So I think uh, this was one of the earlier ones. So this is like to 2010. So one of the earlier ones that got published. And the newer ones, I think, are, sorry, I think this is the latest one. So here they have all, I think 2015. I think in 2022 also there is one, if I'm not wrong. 2022. Huh. This is the 2022 paper. They've expanded it with 600 trios as well. So this is also published in Excel. So yeah, this is what the whole thing. They have done Illumina 30x coverage. Uh, they've detected these kind of variants and these things. So okay, you can give these papers a read. These are very interesting. So you get an understanding how large scale genomics works basically, and that is kind of the what to say. Like people that is what the next 10 years about to happen. So in India also we are doing a project called the Genome India project. So if you are so firstly, this is the Genome Asia project where they sampled 100K samples across Genome Asia. Sorry, Genome Asia 100K is the project name. Around 2,000 samples across Asian countries, which includes India as well. So you can take a look at this as well. The Genome India project is currently ongoing. We do not have the paper yet. This is actually the commentary paper for that. You can take a look. The main flagship paper is coming in some months. So these are all the samples that have been collected from India. So if you want have an interest in genomics, do read these papers. They will give you a greater insight about how genomics works in large scale projects. And also the UK Biobank is a great resource for that as well. Okay, uh, going back. Okay, do you have any answers? These are easy, guys. I, I hope everybody knows, but the answer is motor protein. So the protein will be there. It will detect the change of. So because of that, um, the single-stranded DNA will go down the protein and it will detect the change of ions flowing in and out of the protein. So the answer is motor protein. The answer is basically all of them. Uh, yeah, I have not covered the Ultima Genomics part, but Sir has covered in his lecture, so I think yeah, this should be fine. Yeah, yeah, yeah. So first you tag the base, then you wash it, then you image, then you clean the label, again wash, then you repeat. So option three is the correct one. Not explaining this. Yeah. Now, so basically, if you do sequencing by synthesis, it can lead to molecular scarring because you are synthesizing a molecule, then you are like using a tag on that, and then you're removing the tag. So both of them, and since in Illumina also you use sequencing by synthesis, so molecular scarring can happen. So sequencing by binding and MN sequencing by binding, sequencing by synthesis is basically the way if to not have molecular scarring. Yeah. Okay, I have not spoken about LD, but basically LD is the non-random association of certain genotypes in a region, right? So generally what happens is like if you're getting a part of the genome from one particular parent, the variants in that particular genome will be inherited together, right? So that is the non-random association of these alleles in that particular region. So generally, the recombination rates will also change if the distance increases. So technically, your linkage disequilibrium also should decrease when you are increasing distance. So it is always a curve like this. It's always a decreasing curve, right? So the more you move away, I think hyperbola is still something that can be an answer, but decreasing curve is a better option here because the, the distance that you increase, the rate of recombination also gets higher. The, mm, the probability that those two alleles will be inherited together are going to be lower. So it's a decreasing curve. Wait, the question was which of the following steps are involved in high-C sequencing? I don't think proximity synthesis is included. Uh, I don't think that. Yeah, one second. Let me check. One second. In high C, you use biotin, right? So the affinity labeled uh, part comes from there. One second. Yeah, this is the paper for uh, high C sequencing. If you want to take a look. Proximity ligation, I'm not so sure about that. Affinity labeling definitely happens. Restriction digestion definitely happens. Single end sequence doesn't happen. But proximity synthesis probably not. But I'm not very sure. But affinity labeling definitely happens because you do tagging using biotin, right? So the answer will be affinity labeling and restriction digestion. Yeah. This I think is easier. It basically tells you about, so if you have somehow two regions of the chromosome that are in close proximity and they are like distance wise they are very far apart, but they say, for example, it is very far apart but it folds and forms some sort of enhancer component for this promoter, that those kind of things can happen, and those are the information that are uh, sorry, paired end. Single end does not happen. Paired end happens. Not sure about the proximity synthesis. I don't think so. It happens. Yeah. In this case, this answer is actually both, because it can tell you the proximity to the nucleus as well as proximity across chromosomes. So one part of the chromosome may be close to another, but actually distance-wise they might be very different, distinct. So yeah. Sorry, were you asking this? You're asking about application of high C. High C is basically, as I said, say for example, you have a long stretch of. Are you asking the application of high C, right? Yeah. So basically, say you have a long stretch of DNA, right? So in the DNA, you'll have a gene. Upstream of the gene, there will be a promoter, and then upstream there will be other elements. That is the whole structure of the gene. But say, for example, 500 kb upstream of this particular promoter, there is some sequence of the DNA which actually affects the gene expression. Now, how does it affect? It might, what has happened is during the conformational structure of the, how the DNA is folded, that 500 kb upstream base actually gets folded and gets attached to the promoter here, right? But you will not be able to know until you know that this is the chromosome capture conformation in that particular location, because base wise that is 500 kb upstream. Technically, it should not have to do with anything of a gene expression that is 500 kb downstream of that region, right? But if you do a high C, you can get the how the structure of the chromosome is at that region, and you will see that 500 kb upstream region has folded and is acting as an enhancer to your promoter, and they are in very close proximity, the enhancer and the promoter. But in distance wise terms, it is 500 kb upstream. But since the formation is conformation is like that, it has folded and come back at this place. IC can capture that. Just by sequencing, you cannot tell that that is actually an enhancer for this. Do you understand? It can be anything. It can be enhancer, it can be repressor, it can be anything. It is affecting that gene expression. Technically, it should not. Like 500 kb upstream, that is not part of the gene. Technically, it should not affect the gene expression at all, but it affects. How it affects? Because of the conformation of the DNA at that region. That is how it affects. And that information, that conformational information, will be given by HiC. You cannot do it just by sequencing, because when you do sequencing, you'll see that that region is 500 kb upstream. So by default, you'll assume that it has got nothing to do with the gene expression, right? But if you have the IC data, you capture the conformation of that region, you can tell that actually that might be somehow altering expression here. I hope I was clear. Okay, the answer, so basically Chicago library preparation mainly deals with the biotinylation and basically ligation of the, uh, basically proximity ligation and biotinylation. So nucleosomes are added artificially to mimic that particular region, and biotin labels are added. Streptavidin, I think, is added, and ends are ligated as there are no free ends. So yeah. It is basically the idea is basically that you create a library of DNA fragments through a specific protocol using this ligation and biotinylation and other kind of uh, streptavidin or other molecule addition. I hope that was correct. Yeah, for long read, repeat again. Repeat array assembly is very good for long read. Haplotype phasing is very good because you have longer stretches of DNA. You know which strand the variant is in. So haplotype phasing is also good. Allele unaware de novo assembly, that is not a strong point of LRS, and it is not how LRS is done. SV detection, again, large deletions, large insertion detection, that is also very useful for LRS. I mean, we discussed this while talking about long read sequencing, so this should be fine. Moving on. Yeah, the answer is that longer inserts can be used for continuous long read sequencing over circular consensus sequencing. So again, it's I think uh, it's not more accurate because again, long read sequencing it's scale there are scales with the length of the read. So yeah, I think this is pretty obvious. You guys are not answering, so I'll discuss. I'll not wait for your answers, then I'll discuss and go on. Okay, so yeah, SMRT, the bell formation at the end. So you have, if you remember this thing, this bell formation, smart bell, is basically the signature of SMRT. Yeah, and you can have longer insert size because it's a long read sequencing. So 1 KB to 10, 100 KB insert size and SMRT bell template as well. Okay, for PacBio long read, the DNA polymerase remains fixed because the circular template moves along the DNA polymerase. So you have a fixed DNA polymerase, and the since it's circular DNA, it moves along. So that is the way PacBio long read works, SMRT as well, because of the circular nature of the DNA. This I'll not wait. So enzymeless DNA sequencing is done using ONT because you have to detect the change in bases by changing the current of ion flowing, right? So this is pretty straightforward. Next. Again, this is something either you know or you don't know. So GWAS is basically genome-wide association study. What we do is basically you get all these variants and you also take phenotypes of the individuals that you have sequenced. So you have, say for example, 20,000 individuals with all their variant information, and you have the phenotype information as well. Now do you do a regression kind of a model where presence or absence of a SNP, you check if the phenotype is going up and down. So that is what a genome-wide association study is, and then you get certain variants in certain genes which are statistically significantly associated with these phenotypes, and those are what you report, and those are reported in the GWAS catalog, right? So if you want to take a look at that as well, it's the EBI GWAS catalog. You can take a look here as well. Okay. Okay. I'll also quickly go over this. So variant calling format file, VCF file, I think I showed you last time. So it can contain all type of variant information of a population, right? So it can contain structural variant information, it can contain single nucleotide variant information, it can contain uh, repeat expansions, STRs, and this kind of thing. All type of variations can be present in the VCF format. I think next day, I think probably I'll show you a VC file. I think it'll be useful. And but the answer is that you can store all kind of genetic information, variation information there, not only single nucleotide or SV. You can store any type of, and you can also have annotations, right? You can directly annotate the variants in the VC file and keep it annotated there only. So that is also another advantage. So only the first option is correct. Here again, the answer is African because, yeah, it is how it is seen. If you read the Thousand Genome paper, there are a lot of African samples there. There also they talk about the diversity of African samples compared to European. Europeans will be the most less diverse because due to thousands of years of kind of staying together in breeding and those kind of things, Africans have migrated. So the out of Africa event is the most well-known event uh, in evolutionary history. So we all somehow like originated in Africa and then migrated out of Africa. So the most genetic diversity that we'll find is in African population, not Asian, African. No, by the way, VC file is not a text file. Let me see if I have a VC file. I think I'll have. Yeah, this is how a VC file looks. This is not a text file. So you have chromosome, then position, then ID of the SNP, reference allele, alternate allele, then you have quality, and then you have like these are the samples and what are the genotypes. So zero meaning zero zero. In some cases you'll see one or two. One is basically zero one and two is one one, and it is given like this. The start of the heading starts like this: file format, VCF, whatever, date, and this kind of all the information is captured in this way. VCF is not a text file, by the way. Okay. Haplotype is basically the stretch of the genome that is inherited together, meaning like either it is directly coming from the father or directly coming from the mother. So it is not like half coming from mother, half coming from father. It's the whole region that is coming from one of the parents. That is what the haplotype is. And the SNPs in a haplotype are in complete linkage disequilibrium. Not perfect. Perfect would be like 100% correlation, and not it is not actually 100% in most cases, but it is in complete LD because those are inherited together. Maybe like zero point 0.6, zero point 0.8 is the correlation R square value, but they are not in perfect. Perfect would imply one, right? So that's a separate concept. But complete linkage disequilibrium, definitely because they are inherited together. Okay. Uh, yeah, that is all from my side. Sorry, I took a little bit more of time. Now, perfect LD will be like kind of like every time you have that particular snip, you have the other snip. That is in perfect LD, right? So the R square value you calculate. So okay. So how do you calculate linkage disequilibrium? Basically, one snip is present. The other snip, how many times out of like whatever number of individuals you're checking, say for example, 10,000 individuals you're checking, how many times these two snips are together? If one snip is present in that individual, the other snip is also present. How many times is it? Is it like 10,000 out of 10,000? Then it is in perfect LD because every time this snip is present, the other snip is also present. But complete linkage disequilibrium meaning like the whole haplotype, the region of the genome is inherited together and the SNPs present in that haplotype are in LD with each other. But they not might be in full LD. They might not be every time this SNP is present, the other SNP might not be present. It might be like every 100 times this SNP is present, 80 times this SNP will be present. So that is like a complete LD, uh, what you can call, but perfect LD would be like every time the other snip is also present. That is what we call perfect LD. So that is not the case in case of a haplotype. We consider zero point 0.5, zero point 0.6 R square. R square is basically the correlation coefficient. So every 100 times this particular snip is present, 80 times this snip will also be present. So that they are in LD at zero point 0.8, or if it is 60 times present of 100 times of this, they're in LD of zero point 0.6. So that is how LD is calculated. So if it is 1, every time this snip is present, this snip is also present, then it will be in perfect LD. But it is like haplotype is not defined that way. Okay. Any more questions? If not, I'll close the session because we are already running 15 minutes late. Okay. Okay. Thank you. Thank you for joining. See you next week, Tuesday, same time. Thank you.